Welcome to the deep dive. We're here to plunge headfirst into some really complex topics so you can surface feeling well informed.
Glad to be here.
Today, we are tackling something you almost certainly have at home: acetaminophen.
Yeah, or paracetamol, depending on where you are.
Yeah.
APP for short, maybe.
Right. APP is probably the most common pain reliever, fever reducer, on the planet.
It really is. The exposure levels are just enormous, especially when you think about pregnant women and young children.
Do we have numbers on that? Like, how common is it?
Oh, yeah. Surveys show, I think, over 65% of pregnant women in the U.S. use it at some point. And it's over 50% in Europe, too.
Wow. Okay.
And for decades, it's been seen as the safe option, especially during pregnancy, early childhood. Safer than NSAIDs, less acute risk.
Okay, so that's the background. Generally considered safe, but there's always a but, isn't there?
There usually is when you dig deep.
Yeah.
Despite this widespread acceptance, some recent research has started raising some, well, pretty uncomfortable questions.
About what specifically?
About its long-term safety, particularly its impact on neurological development. We're starting to see potential links being drawn between APAP exposure and the, frankly, startling rise in things like ADHD and autism spectrum disorder, ASD.
And that rise is startling. I remember seeing figures like for ADHD in U.S. kids aged 4 to 17.
Exactly. It jumped significantly, went from, 7% up to over 10% in just a couple of decades.
That's a huge increase.
It is.
Yeah.
And autism prevalence, too, it's climbed dramatically. Now, some of that is better diagnosis, broader criteria, sure.
But not all of it.
Not all of it. A change that rapid, you just can't explain it by genetics alone. The human genome doesn't shift that fast. It points pretty strongly towards, environmental factors, exposures happening during really critical developmental times.
So that's our mission for this deep dive, then, to really sift through the evidence.
Precisely. We've got a whole pile of sources here, big epidemiological studies, tracking thousands of moms and kids over years.
And not just self-reports, right? There's biomarker data.
Yes, objective biomarker assessments, that's crucial. Also, ecological analyses looking at population trends, and then mechanistic stuff from animal models trying to figure out the how.
Okay. So we're examining everything from prenatal, meaning during pregnancy, through perinatal, around birth, and postnatal, so early childhood exposure.
Right. And the core paradox we need to explore for you listening is this gap. You've got the medical community generally saying it's safe, based on older data maybe. But then you have this growing body of newer studies suggesting that actually the developing brain might be incredibly sensitive to APAP, especially during these key windows, say from the first trimester right through the first few years of life, maybe up to age 4 or 5.
Okay, let's dive into that first window then, the prenatal link. What are these big human cohort studies telling us?
Well, the first thing that jumps out is the consistency. It's not just 1 or 2 studies. We're talking multiple large cohorts, like the Danish National Birth Cohort, the DNBC, that's a huge one, and others across different countries.
And
They're consistently finding a positive association between mothers reporting using APP during pregnancy and certain adverse outcomes in their children.
particularly what kind of outcomes?
The link seems strongest and most consistent when it comes to attention-related issues. ADHD symptoms, hyperactivity, difficulties with executive function.
Okay.
Several studies, using different questionnaires or even clinical diagnoses, they land on this same positive association. The size of the effect, the risk increase, it's often modest, maybe not gigantic.
But it's repeatable.
It's repeatable across different populations. That's the key.
Do we have a specific example just to frame the level of risk we're talking about?
Yeah, sure. One Danish study, looking specifically at autism, found that prenatal APA use was associated with an increased risk of autism with hyperkinetic symptoms. That's often seen as a more severe form, kind of overlapping with ADHD.
Yeah, and the number?
The hazard ratio, or HR, was 1.51.
Okay, let's translate that. A hazard ratio of 1.51 means the group exposed to APAP during pregnancy had a 51% higher rate of developing that specific outcome compared to the group that wasn't exposed.
Exactly. It's a 51% increased risk. It's statistically significant and certainly enough to make you pay attention.
And it wasn't just that specific subtype. There were links to ASD generally, too.
Yes. Studies indicate links to ASD risk overall, although, as I said, the connection often seems clearer, maybe more consistently found, for the ADHD or hyperactivity components.
Now, you mentioned duration earlier. Does it matter how much or for how long a mother takes APA during pregnancy?
It absolutely seems to. The effect isn't just binary, like yes-no exposure. It appears to follow a dose-response pattern.
Meaning more exposure, more risk.
Right. Specifically, prolonged use seems to be the bigger concern. Some studies defined long-term as, say, using APAPE for more than 28 days total during the pregnancy.
Okay, 4 weeks or more.
Yeah. And that longer duration was found to be much more strongly associated with these adverse neurodevelopmental outcomes compared to just short-term use, like for a day or two.
Which makes sense biologically. Cumulative exposure. But wait a minute. If someone's taking APAPE for over 28 days during pregnancy,
Hmm.
doesn't that suggest maybe there's a serious underlying issue? Like chronic pain, a persistent infection, something else going on that's driving the APAP use? How do we know it's the drug and not the underlying condition?
That is the absolute million-dollar question in these observational studies. It's called confounding by indication. The reason for taking the drug might be the real risk factor, or part of it.
So how do researchers handle that?
They try to adjust for it statistically.
Yeah.
They collect data on why the mother reported taking APAP. Was it fever, infection, headaches, muscle pain, preeclampsia maybe? And they factor that into their models.
And does the link disappear when they do that?
Often, no. Even after adjusting for the self-reported reason for use, the association between APAP and the neurodevelopmental outcome frequently persists.
Interesting. So that leans more towards the drug itself playing a role, not just the illness.
It suggests that, yeah, it makes it harder to dismiss the drug as irrelevant. But there's another issue with these early cohort studies.
Which is?
Many relied on self-report. Asking a mother, maybe months or even years later, to remember how often she took a common over-the-counter drug like APAP during her pregnancy.
That sounds really difficult, prone to errors.
Exactly. There's recall bias. You might forget taking it for a minor headache 9 months ago.
Yeah.
And reporting bias, maybe you worry about admitting you took medication. It's just not perfectly reliable.
So how did the research move beyond that?
That's where objective measures came in. And it really strengthened the evidence. One key study, based in the Boston birth cohort, didn't just rely on memory.
What did they do?
They measured biomarkers. Specifically, they looked for metabolites of APAP in the mother's blood plasma, collected objectively right after delivery, like 1 to 3 days postpartum.
Okay, so that gives a snapshot of recent exposure levels based on actual chemistry, not memory.
Precisely. A much more reliable indicator of exposure around the time of birth, at least.
And what happened when they used those objective biomarkers instead of just asking the mothers?
The findings are pretty striking. They found significant positive associations between the levels of these AP metabolites in the mother's blood and a later diagnosis of ADHD in her child.
And was it dose response again?
Yes, crucially, it was dose responsive. Higher levels of the metabolite measured at delivery correlated with higher odds of the child having an ADHD diagnosis down the line.
Wow.
And importantly, just like the other studies, they confirmed these links held up, even after adjusting for the indication the reason the mother might have taken the drug, like fever or pain.
And was it specific to ADHD?
In that particular study, yes, the association seemed most robust for ADHD compared to other neurodevelopmental disorders they looked at. That specificity adds weight to the idea that APP might be interfering with particular pathways relevant to attention and hyperactivity.
Okay, this is getting compelling. But the critics will still bring up confounding, right? Maybe women who take more AP are just different in other ways. Maybe they have underlying genetic risks for psychiatric issues themselves, which they pass on to the child.
Absolutely. That's always the challenge. Researchers do adjust for a lot. Maternal age, BMI, smoking, alcohol, even pre-pregnancy psychiatric history. But the gold standard for trying to eliminate that shared family risk, both genetic and environmental, is the sibling comparison design.
Ah, yes, the sibling studies. Explain how those work again.
You find families with at least 2 children. You compare a child who is exposed to APAP in utero to their sibling who wasn't.
Same parents, same home environment, roughly half the same genes.
Exactly. So theoretically, many of those shared genetic and background environmental factors that could confound the results are controlled for inherently within the family unit. Any difference in outcome between the exposed and unexposed sibling is less likely to be due to those stable family factors.
That sounds like a really powerful way to test the hypothesis. So what did these sibling studies, mostly out of Norway, I think, find? Did the link hold up?
Well, this is where it gets a bit messy. The results were actually conflicting.
Oh, how so?
Some sibling studies, especially the larger ones, or those that use continuous measures of, say, hyperactivity symptoms rather than just a yes-no diagnosis, did still find significant associations. The link persisted even in the sibling comparison.
Okay.
But other sibling studies, sometimes smaller ones, or those focusing only on a clinical ADHD diagnosis as the outcome, found no statistically significant link. The association seemed to disappear in those analyses.
Why the difference? Did the sibling control just get rid of the apparent effect, proving it was confounding all along? Or is there something else going on?
It could be a mix of things. Statistical power is one issue. Smaller studies might just not have enough power to detect a real but maybe modest effect. Methodological differences matter too. But there's a more subtle point to consider about sibling designs. While they're great at controlling for stable shared factors, they might accidentally eliminate the very thing that makes APAP risky in some families.
How does that work?
Imagine if APAP only causes significant harm when it interacts with a specific shared family vulnerability, maybe a genetically slightly less efficient detoxification pathway, like impaired sulfation, or naturally lower baseline glutathione levels.
Okay.
The sibling design, by controlling for shared family factors, might actually be adjusting out that crucial shared vulnerability. So you might conclude APAP has no effect in that comparison when actually it does have an effect, but only in the presence of that shared familial trait that the design removed from consideration.
That's a really interesting point. The control itself might hide the interaction.
It's a potential bias, yeah.
Yeah.
It means a null result in the sibling study doesn't automatically mean APAP is completely off the hook. It just might mean the risk isn't universal, but contingent on other factors that run in families.
Before we move off the prenatal period, what about sex differences? We know ASD, for example, is much more common in boys. Did the studies see different effects in boys versus girls?
Yes, absolutely. And it's not always straightforward. Some outcomes, like maybe attention scores measured by specific tests in a clinic, seem similar for boys and girls after prenatal exposure. But for other measures, especially things like parent-reported executive function difficulties, the effect estimates were often significantly higher for boys. The risks seemed greater for them.
Okay, so boys potentially more vulnerable for certain outcomes.
Potentially. But then other studies found female-specific effects. One Swedish study, for instance, linked prenatal APAP exposure specifically to language delay, but only in girls around 30 months old.
So it's complex. The risk profile isn't the same for males and females, suggesting whatever APAP is doing, it's likely interacting with sex-specific developmental processes or hormones.
That seems very likely, yes.
Yeah.
It adds another layer to the biological plausibility question we need to get into.
Okay, let's shift focus then. We've talked prenatal. What about after the baby is born? The postnatal period, infancy, is that still a critical window?
Oh, absolutely. You could argue it's an even more sensitive period in some ways. The brain is undergoing explosive development, forming crucial connections, and it doesn't have the same protections as an adult brain. The blood-brain barrier is still maturing, detoxification pathways are immature.
And what does the animal research suggest about APAP exposure during this early infancy stage?
Preclinical models, mostly in rodents, suggest that giving even near therapeutic doses of paracetamol in the first few days of life can lead to some profound, long-lasting neurological changes.
And is the mechanism different in newborns compared to adults?
It seems to be. In newborns especially, the liver pathways for metabolizing APAP, like glucuronidation and sulfation, aren't fully developed. This might mean more of the drug gets shunted down that alternative pathway, creating the toxic metabolite, NAPQI.
Ah, NAPQI again.
Right. And because the liver isn't fully equipped, and maybe the blood-brain barrier is leakier, the brain itself might become the primary target for toxicity, rather than the liver damage we typically see in adult overdoses. This makes that immediate postnatal period uniquely vulnerable.
And does the human observational data back this up? Does the risk change if the exposure happens after birth instead of during pregnancy?
It looks like it might. Some observational data suggest that postnatal exposure, meaning giving APAP to the infant or toddler in their first year or two, might carry an equal, or possibly even a higher, risk of developing autism compared to prenatal exposure alone.
Higher risk. That's significant.
It really underscores the idea of this postnatal sensitive window, which makes measuring exposure during that time really important but also really challenging.
How do researchers even track that? Parents might give doses here and there for teething, fevers, colds. It's not like a prescription.
They've tried different approaches. Some cohort studies simply interviewed mothers periodically, asking about any medication use in the child, maybe up to 18 months of age. If the child received any APP dose in that window, They were classified as exposed.
Okay, that's pretty broad. Any Nord.
Right. A more detailed approach came from a survey-based study. They actually asked parents to quantify the exposure, to estimate the number of doses their child received before turning 2.
How specific did they get?
The answers were categorized into ranges,
Right.
from none up to something like 64 or more doses.
Wow, 64 plus doses before age 2. That implies pretty regular, maybe chronic use for some kids.
It does. And when they used this more quantitative measure of postnatal exposure, they came up with a truly staggering statistic.
Which was?
They estimated the population attributable fraction. That's like the percentage of cases in the population that could theoretically be linked to that exposure factor.
Right.
For postnatal APAP exposure, they estimated it could account for approximately 40% of the risk of an ASD diagnosis, specifically among male children in the U.S.
Hold on. 40%. 40%.
That's enormous. If a single common medication could potentially be linked to 2/5 of male autism diagnoses, that completely reframes the safety discussion for early childhood use.
It absolutely does. It's a number that's hard to ignore, even with the caveats of survey data. And it leads us directly into another really striking piece of evidence, this ecological correlation involving circumcision.
Circumcision. How does that connect to APAP and ASD?
Well, first, remember the established fact. ASD is diagnosed about 4.6 times more often in boys than in girls.
Right, the sex bias is well known.
Okay, so researchers looked at country-level data. They compared the rates of male circumcision in different countries with the prevalence rates of ASD in boys, specifically boys born after a certain year.
And what did they find?
An incredibly strong correlation, almost a perfect linear relationship. The correlation coefficient, R, was 0.98. 0.98, that's nearly a perfect match. What was the cutoff year?
Boys born after 1995.
1995. Why that specific year? That seems too precise to be random.
It's not random at all.
Yeah.
1995 marks a major shift in medical practice regarding pain management for newborn circumcision.
Ah, okay. What changed?
Before the mid-90s, believe it or not, circumcision was often done with little or no pain relief. There was this outdated and completely wrong idea that newborns didn't really feel pain the same way.
That's horrifying.
It is. But research in the early 90s finally made it clear that infants absolutely feel pain and need analgesia. So the guidelines changed.
They started recommending pain relief.
Yes. A key study came out in 1994 showing that giving paracetamol regularly, like every 6 hours for the first 24 hours after the procedure, significantly reduced the baby's pain responses.
So APAP became the standard of care.
It largely did. Guidelines from organizations like the American Academy of Pediatrics were developed and widely adopted. They often recommended giving a dose before the procedure, then repeated doses every 4 to 6 hours for about 24 hours afterwards.
Okay, so suddenly, starting around 1995, a huge number of newborn males undergoing this common procedure began receiving a predictable pattern of maybe 5, 6, 7 doses of APAP right in their first few days of life.
Exactly. Right during that period we just discussed as being potentially hyper-vulnerable neurologically. This widespread standardized change in practice, driven by the very necessary goal of pain management, created this sudden population-level spike in early life APAP exposure, specifically for males.
And the timing of that practice change correlates almost perfectly with the rise in male ASD prevalence in those countries.
The ecological data shows it extremely strong, R equals 0.98 correlation. Well, correlation doesn't equal causation, of course. It provides a really plausible potential mechanism contributing to that stark 4.6 to 1 male bias in ASD diagnoses. It's hard to dismiss.
One more connection before we get into the biology, the link between APAP and vaccines. This is a really sensitive area because of misinformation.
It is, and it's important to be clear. The issue isn't the vaccines themselves. Vaccines are crucial public health tools. But historically, and even sometimes now, parents were often advised to give APAP either before or after vaccinations, just in case the child developed a fever or soreness.
Prophylactically, right.
Right. So if a child received APAP around the time of vaccination and later developed signs of autism, some parents, understandably but incorrectly, might draw a link between the vaccine and the autism.
When the real issue might be the APAP given during that period.
Potentially, yes.
Well,
Or more broadly, the issue is giving APAP during any period of inflammation or immune activation, whether it's post-vaccination fever or fever from an illness. We know immune activation itself can be a vulnerable time for the developing brain. Adding APAP with its own metabolic effects we're about to discuss might act as a second hit or a compounding factor during that already sensitive inflammatory state.
okay, that distinction is critical. It's not the vaccine, it's potentially the medication given alongside the immune response.
Exactly. It highlights the problem of widespread APAP use during these common periods of childhood inflammation.
All right, let's get into the how. We've seen the correlations in humans, the ecological links. What's the biological story? What could APAP actually be doing in the body, especially the developing brain, that might cause harm? Starting with this pathway involving glutathione.
Okay, so APAP metabolism 101. Most of it gets processed safely in the liver through two main pathways, glucuronidation and sulfation. These create inactive products the body can easily get rid of.
Fine so far.
But a small percentage, maybe around 5% normally, goes down a different route. It gets metabolized by enzymes in the P450 system. And this creates a byproduct called NAPQI and acetylphenylketonuria.
And NAPQI is the bad actor here.
NAPQI is highly reactive, highly toxic. It wants to bind to things in the cell, especially proteins, and damage them.
So why does the body even make it?
Well, it has the defense mechanism. Normally, NAPQI is immediately snatched up and neutralized by the body's master antioxidant, glutathione, usually abbreviated as GSH.
Okay, GSH saves the day.
It does, by conjugating with NAPQI and making it harmless and easy to excrete. The problem comes when that system gets overwhelmed.
How does that happen?
Two main ways. One, if the sulfation pathway, one of those main safe routes, isn't working well. And interestingly, impaired sulfation capacity is something that's been observed pretty commonly in kids with autism.
Okay, so one safe route is potentially compromised already in some individuals.
Right. The second way is if your baseline glutathione, GSH stores, are already low, maybe due to chronic illness, poor nutrition, or other environmental toxin exposures.
So low GSH means less defense against NAPQI.
Exactly. When GSH is low, or when you take a large dose of APAP creating a surge of NAPQI, the GSH gets used up trying to detoxify it. If you run out of GSH, the NAPQI is free to cause damage, binding to liver cells, and crucially, potentially brain cells, especially in vulnerable neonates.
And this depletion of glutathione, is that considered significant in the context of autism?
Many researchers point to chronic or repeated GSH depletion as a highly plausible contributing factor, maybe even a single key mechanism in autism. Because GSH isn't just for detoxifying APAP, it's crucial for handling all sorts of other harmful agents like heavy metals, pesticides, oxidative stress byproducts.
So depleting GSH with APAP makes the body and the brain less able to cope with everything else.
Precisely. It lowers the body's overall detoxification capacity. And we have animal studies that back this up specifically for APAP. Prenatal exposure in rodents led to measurable decreases in GSH levels right in the hippocampus of the offspring, particularly males. They also saw decreased activity of another key antioxidant enzyme, SOD, superoxide dismutase.
Okay, so the mechanism is there in the animal models too. Lower antioxidants in key brain regions.
Yes, directly measurable.
Now, how does this GSH depletion connect to other biological issues often seen in ASD and ADHD, like problems with energy, metabolism, or mitochondria?
That's a direct link. When GSH is depleted, the cell can't effectively neutralize reactive oxygen species free radicals. This leads to increased oxidative stress. Think of it like cellular rust.
Then mitochondria are vulnerable to this.
Extremely vulnerable. Mitochondria are the cell's power plants, generating energy. Oxidative stress damages them, leading to mitochondrial dysfunction. They can't produce energy efficiently.
And mitochondrial problems are common in neurodevelopmental disorders.
Yes, it's a recurring finding. Researchers observe abnormalities in mitochondria in individuals with ASD and ADHD, things like changes in the amount of mitochondrial DNA, reduced efficiency of the energy production machinery, particularly noted in brain regions like the frontal cortex.
So APAP depletes GSH, leads to oxidative stress, which damages mitochondria, potentially impairing neuronal function.
That's the chain of events, yes. And again, studies in APAP-exposed mice support this. Gene analysis showed that genes involved in responding to DNA damage and in glutathione metabolism were upregulated, basically. The cells were scrambling to deal with oxidative stress hitting their DNA.
Okay, that covers the toxic byproduct route. But what about APAM's intended effect? How it actually reduces fever and pain? Could that process itself be involved? You mentioned prostaglandins earlier.
Right. This is another really important potential mechanism. APP works, in large part, by inhibiting an enzyme called cyclooxygenase 2, or COX-2.
COX-2. And what does COX-2 normally do?
COX-2 produces signaling molecules called prostaglandins. Prostaglandin E2, or PGE2, is a key one. It's involved in generating fever and inflammation signals, which is why inhibiting it reduces those symptoms.
But PGE2 does other things too, especially in the developing brain.
Crucially, yes,
Yeah.
PGE2 isn't just about fever. It plays vital roles in normal brain development. It helps guide neurons to their correct locations. It's involved in synaptic plasticity, the basis of learning and memory, and, really importantly for the sex differences we see, it's involved in the masculinization of the developing brain.
Wait, masculinization. So blocking PGE2 could interfere with typical male brain development.
That's the hypothesis, based on animal work. By blocking PGE2 synthesis with APAP during critical periods, you might be inadvertently disrupting these essential developmental processes.
Is there specific evidence for this?
Yes. Studies in rats show that disrupting PGE2 synthesis altered the development of Purkinje cells in the cerebellum.
The cerebellum that's involved in coordination, but also attention and social behavior, right? And heavily implicated in autism.
Exactly. And in this rat study, inhibiting PGE2 led to cerebellar atrophy, basically shrinkage and altered social interaction behaviors later in life, but only in the male rats.
Only in males. That lines up perfectly with the higher ASD rates in human boys.
It provides a very specific, biologically plausible mechanism linking APIP's known action, PGE2 inhibition, to a sex-specific effect in a brain region known to be affected in ASD. It's quite compelling.
Beyond these structural and hormonal effects, does APIP affect other key brain molecules like growth factors?
It seems so. There's evidence linking APAP exposure to changes in brain-derived neurotrophic factor, BDNF.
BDNF. I know that's critical for neuron survival, growth, making connections.
Absolutely essential. And mice treated with APAP showed altered BDNF levels in important brain areas, like the frontal and parietal cortex regions, vital for executive function, planning, sensory integration. Lower or dysregulated BDNF could certainly impair healthy brain wiring.
And you mentioned epigenetics earlier. APIP changing how genes are expressed?
Yes, there's evidence for that too. APAP exposure seems to affect DNA methylation patterns. Methylation is like little chemical tags on DNA that tell genes whether to be switched on or off without changing the actual DNA code itself.
So APAP could be reprogramming gene activity.
Potentially. Studies have shown altered methylation on genes crucial for brain development and function after APAP exposure. Genes involved in managing oxidative stress, in neural signaling, and synapse formation. Key names like MECP2, RELN, BDNF itself, all genes strongly linked to neurodevelopmental disorders. So APAP might be exerting subtle, long-lasting effects by changing the operating instructions of these critical genes.
And are there even broader effects beyond the brain, like on hormones or the immune system during pregnancy?
Yes, the picture suggests APP isn't just acting locally. Some data indicates it might function as an endocrine disruptor during pregnancy. Studies in animals showed it could affect how the mother's immune system adapts to the pregnancy, potentially decrease maternal progesterone levels.
Progesterone is vital for maintaining pregnancy and fetal growth, right?
Absolutely. So effects like these suggest APA could have systemic toxicity, hindering fetal development in ways that go beyond just direct brain effects or liver toxicity. It paints a picture of a drug with potentially wider biological impacts than often assumed.
Okay, so we have multiple plausible biological mechanisms. GSH depletion leading to oxidative stress and mitochondrial damage, disruption of essential PGE2 signaling affecting brain structure and masculinization, altered BDNF epigenetic changes. It's quite a list.
It is. It shows there are several biologically coherent ways APA could interfere with neurodevelopment.
But we know these disorders are complex. It's rarely just one thing. How might APAP interact with other common childhood factors? You mentioned an antibiotic, ear infection, glutamine triad. Let's unpack that.
Right. This looks at the synergy between common events. First, we see in studies that postnatal antibiotic use and recurrent ear infections are both independently associated with a higher risk of ASD, and they often go together, right?
Right.
Ear infection leads to antibiotic prescription.
Makes sense. The correlation is strong.
Estimated around 0.64 between antibiotic use and ear infections in some data sets. Now think about what broad-spectrum antibiotics like amoxicillin clavulanate, which is very common, do to the gut microbiome.
They wipe out a lot of bacteria, good and bad.
Exactly. And some of those gut bacteria are crucial for synthesizing essential amino acids, including methionine.
Methionine, why is that important here?
Methionine is a precursor building block for making sulfate, which we need for that safe sulfation detoxification pathway. And it's also crucial for making glutathione, GSH.
Ah, So antibiotics might reduce the raw materials needed to make GSH. At the same time, APAP is using up the existing GSH stores.
That's the concern. It's like a double whammy on the body's detoxification capacity. And this ties into something called the glutamine
The
paradox.
glutamine paradox.
Glutamine is another amino acid needed for GSH synthesis.
Hmm.
But researchers consistently find that plasma glutamine levels are often low in children with ASD, even though you'd think the body would need more. It's a paradox.
Why might that be?
It's complex, but this low glutamine might further limit GSH production, and it has other knock-on effects. Low glutamine can limit the availability of another amino acid, arginine.
Arginine. What does that do?
Arginine is needed to make nitric oxide.
Nitric oxide. The vasodilator.
Exactly. Nitric oxide helps relax blood vessels, ensuring good blood flow, including to the brain. If glutamine depletion limits arginine, which limits nitric oxide, you could potentially compromise cerebral blood flow, maybe contributing to a low-level hypoxic state that could further stress those already vulnerable mitochondria. It's another layer of potential synergistic toxicity.
Wow. Okay. So ear infections lead to antibiotics, which disrupt the gut, potentially limiting GSH precursors, while AP depletes GSH directly, and maybe downstream effects impact brain blood flow. It's a tangled web.
It really highlights how multiple, seemingly unrelated common childhood events could interact to increase vulnerability.
And this interaction isn't just limited to other medications, right? What about environmental chemicals? There was mention of glyphosate, the herbicide.
Yes, this is another area of research looking at co-exposures. Some scientists have proposed that the massive increase in glyphosate use, especially on genetically modified crops since the mid-90s, might be another environmental factor contributing to the autism epidemic.
What's the proposed mechanism for glyphosate?
Similar to antibiotics, actually. Glyphosate is thought to inhibit specific enzymes, particularly in gut bacteria, that are needed to synthesize certain essential amino acids again, including methionine and also tryptophan.
Okay, so glyphosate also potentially limits methionine needed for GSH. What about tryptophan?
Tryptophan is the precursor for serotonin, a crucial neurotransmitter for mood and social behavior, and also for melatonin. Reduced tryptophan could mean reduced serotonin and melatonin production, both of which have been implicated in neurodevelopmental issues.
So again, an environmental factor potentially depleting the very resources the body needs to buffer against toxins like APAP's metabolite or disrupting key neurotransmitter pathways.
That's the hypothesis. It suggests a scenario where multiple environmental hits synergize. You might have APAP depleting GSH, while antibiotics and glyphosate are limiting the ability to replenish GSH and other crucial molecules.
Is there any direct evidence of this synergy, maybe showing reversal?
There was a compelling case study involving triplets. All three had signs of autism and also showed high levels of biochemical markers indicating mitochondrial damage, plus high levels of glyphosate in their urine, likely from their diet.
Okay.
They were switched to a strict organic diet, drastically reducing their glyphosate intake. Their urinary glyphosate levels plummeted like a 94% reduction.
And what happened to their symptoms?
According to the report, their markers of mitochondrial damage improved, and their autism signs, particularly those related to mitochondrial function, also showed noticeable decline. Now, it's just one case study, not definitive proof.
But highly suggestive of that interaction between environmental chemical load and underlying metabolic vulnerability.
Exactly. It fits the picture.
Okay, shifting back to human factors, we have to address the elephant in the room.
Yeah.
Parental history. If a parent has ADHD or depression, isn't the child already at higher genetic risk for neurodevelopmental issues, regardless of medication use? How much of this is just shared genetics?
That's a huge factor and a major source of confounding. Large population studies consistently show that if either parent has a diagnosed psychiatric disorder, depression, anxiety, ADHD, etc., the child's risk of ASD is significantly higher.
Is the risk equal from both parents?
Interestingly, no. The risk seems higher if the mother has a psychiatric history. The hazard ratio might be around 2.12 compared to if only the father does, maybe HR 1.63.
And if both parents are affected?
The risk is highest then, as you'd expect. The hazard ratio can jump to something like 3.61. So yes, genetics and the shared family environment associated with parental mental health are clearly powerful drivers of risk.
So how does that connect back to APA use specifically?
Here's the direct link. Studies using polygenic risk scores for ADHD, basically a measure of someone's overall genetic loading for ADHD based on many small genetic variants, found that a mother's ADHD genetic risk score correlates with her likelihood of using APAP during pregnancy.
So women genetically predisposed to ADHD are also more likely to take APAP-P.
That's what the data suggests. There could be various reasons. Maybe ADHD symptoms lead to more stress or physical discomfort. Maybe related impulsivity affects medication choices. We don't know for sure. But it means the underlying genetics might be driving both the maternal condition and the exposure we're interested in.
Which makes it incredibly difficult for observational studies to perfectly separate the effect of the drug from the effect of the shared underlying risk factors.
Extremely difficult. It's the core challenge.
Given all these interacting risks, APP, antibiotics, maybe glyphosate, parental history, does the research offer any hints about protective factors, things that might reduce the risk?
Yes, there are a few signals in the data, all often preliminary. Prenatal folate supplementation is one.
Folate, like in prenatal vitamins.
Exactly. Adequate folate intake during pregnancy has been linked repeatedly to a decreased risk of ASD. Some evidence suggests it might even help buffer against the negative effects of other risk factors, possibly by supporting those crucial methylation pathways we talked about.
Okay, that's important. Anything else?
Breastfeeding seems potentially protective too. Studies have found that early weaning, defined as never being breastfed, or only for a short period like 3 months or less, was associated with an increased risk of ASD.
So breastfeeding might offer some protection.
It might, perhaps through providing key nutrients, supporting a healthy gut microbiome in the infant, transferring maternal antibodies. The mechanisms aren't fully clear, but the association suggests a potential benefit of longer breastfeeding duration.
Okay, let's try to bring all this together. We've covered a lot of ground. If we synthesize the human observational data, the cohorts, the biomarker studies, what's the main takeaway for the listener?
I think the main takeaway from the human data is the consistency. You have, 16 or more decent quality studies now, and they consistently point towards an association between APAP use, especially prenatal and maybe early postnatal, and adverse neurodevelopmental outcomes.
Particularly for attention, hyperactivity, executive function.
Yes, those seem to be the most robust findings. And the fact that the high-quality studies using objective biomarkers found strong dose-responsive links, specifically for ADHD, even after adjusting for indication. That's pretty compelling evidence that there's a real relationship there. It's hard to explain away solely by confounding, though confounding definitely plays a role.
Right. But now we have to throw in the wrench. The animal studies. If the human data looks so consistent, why does the preclinical work often seem to contradict it, or at least muddy the waters? Let's talk about the quantitative weight of evidence review, the QOE.
This is the crucial counterpoint, and it's often cited by those defending APAP safety record. The QOE analysis took a very rigorous approach, setting strict criteria for what counts as a high-quality, relevant animal study.
And their conclusion.
Their overall conclusion, based on the studies meeting their criteria, was that there was no consistent evidence of developmental neurotoxicity, DNT, in rodents exposed to APAP at doses considered therapeutic or non-systemically toxic.
No consistent evidence? That sounds like a flat contradiction to the human studies. How can that be?
It does sound like it, but the devil is in the details, particularly regarding dosage and relevance. The QOE analysis noted that for endpoints like social behavior, stereotyping, attention things relevant to ADHD, the studies that did report adverse effects often had methodological weaknesses or weren't considered biologically relevant or reliable by their strict criteria.
What about the dose? That seemed key.
Hugely key. The QOE review emphasized that in rodents, liver toxicity, hepatotoxicity, happens relatively easily and is a sensitive marker for overall systemic toxicity. They argued that many of the animal studies reporting DNT use very high doses of APAP, often over 200 milligrams per kilograms per day.
And how does that compare to human therapeutic doses?
That 200 millikilogram a day in a rodent, when you try to translate it, is roughly 3.5 times higher than the maximum recommended therapeutic daily dose limit for humans.
Ah, so they were essentially poisoning the animal systemically, causing liver damage and widespread stress, and then seeing neurodevelopmental effects.
That's the argument, that the DNT observed in those high-dose studies might be a secondary consequence of general systemic toxicity, rather than a specific effect occurring at doses relevant to typical human therapeutic use. When they looked only at studies using lower, non-hepatotoxic doses, the evidence for DNT was inconsistent or absent.
So the animal models might not be sensitive enough, or maybe they metabolize APP differently, so the safe dose window is wider for them.
That could be part of it. Rodents might have more robust detoxification pathways, particularly in the developing brain, perhaps higher baseline GSH or enzyme activity compared to humans, especially humans with those underlying metabolic vulnerabilities. The developing rodent brain might just be better equipped to handle the NAPQI metabolite at therapeutic equivalent doses.
So this leaves us with a really nuanced picture. Strong, consistent correlations in complex human populations showing dose response versus carefully controlled animal studies that don't consistently show the same direct neurotoxicity at safe doses, potentially due to metabolic differences or the difficulty of modeling human complexity.
Exactly. Human studies capture the real world. Prolonged exposure, individual metabolic differences like those GSH or sulfation issues, co-exposures to antibiotics, infections, environmental chemicals,
Yeah.
factors that are incredibly hard to replicate perfectly in a standardized animal model. Humans aren't living in controlled lab cages,
Right.
and human exposure might happen during very specific narrow windows of vulnerability that are hard to pinpoint and test systematically in animals, especially when combined with things like fever or inflammation, which aren't always included in the animal protocols.
So where does this leave us regarding clinical advice? If the human evidence suggests caution, but the animal data is less clear at therapeutic doses, why hasn't there been a more dramatic shift in guidelines?
Well, partly it's because the widespread belief in APAP safety is deeply ingrained, often based on those older studies that focused almost exclusively on liver toxicity and simply didn't look for subtle, long-term neurodevelopmental effects. They weren't designed to find them.
So the goalposts for safety testing might have been in the wrong place.
Arguably, yes. When you put the consistent human observational data, the strong biomarker findings, and the multiple plausible biological mechanisms together, the GSH depletion, the PGE2 disruption, it really does point towards a need for, at minimum, much greater precaution. A serious reevaluation of the routine use, especially long-term use, during pregnancy and maybe those first couple of years of life seems warranted. Urging shorter durations, lowest effective doses, that seems prudent based on the current balance of evidence.
Okay, this has been a really thorough deep dive. We've seen a strong, consistent signal in human studies, linking APAP exposure in those critical prenatal and early postnatal windows to increased risk, especially for ADHD and ASD traits.
Yeah, and it's not just theoretical. It's backed by data from thousands of people, multiple countries, and strengthened by objective biomarker measurements.
So if we had to boil down, what are the absolute key takeaways for you, the listener?
I'd say three main things. First, the dose-response relationship for prenatal use seems clear. Longer exposure equals higher risk. Caution with prolonged use is key.
Okay, second.
Second, that early infancy period, maybe the first year or two, but especially the first few days, looks like an incredibly sensitive window. The ecological data around the circumcision guideline change is a stark illustration of that potential vulnerability.
And third.
Third, the biological how is plausible. It's not magic. Mechanisms involving glutathione depletion, oxidative stress, mitochondrial function, and the disruption of essential developmental signals like PGE2 provide coherent biological pathways for potential harm.
What really stands out to me wrapping this up is that APAP's known therapeutic action inhibiting prostaglandins like PGE2 targets, a process we now know is fundamental for brain development, especially male brain development and synapse formation. Yet, its historical safety clearance seems to have relied heavily on just making sure it didn't cause immediate lethal liver failure rather than looking for these more subtle long-term developmental impacts.
Which leads to a bigger, maybe more provocative thought to leave people with.
Go on.
If the safety assessment for a drug as ubiquitous as acetaminophen perhaps missed the mark by focusing too narrowly on acute toxicity, what other widely accepted over-the-counter medications might have unquantified long-term developmental effects, simply because we haven't looked for them in the right way.
Are we using the right screening tools for developmental neurotoxicity for all drugs used commonly during pregnancy or infancy?
Exactly. Which existing medication guidelines might need a fresh look, specifically through the lens of potential impacts on brain development, especially for drugs that mess with fundamental systems like inflammation, hormones, or key metabolic pathways. That's something for you to think about maybe the next time you reach for that medicine cabinet, and consider the broader picture of how we evaluate drug safety in vulnerable populations.